All right, good morning, everyone. We're sorry for that technical difficulty. My name is Anisha Madhav. I'm a technical support scientist here at ACD. I'm joined here today with another technical support colleague of mine, Adeola Adeyemo. I will begin today by first introducing an overview of the RNAscope technology and a detailed RNAscope workflow. Adeola will then walk you through the best practices, troubleshooting, image acquisition, how to go through data analysis, and various resources that are available to you to get your RNAscope up and running. The RNAscope assay is an in situ hybridization technique that utilizes a unique double Z target probe design followed by a single amplification system, which allows you to visualize a single mRNA transcript as a punctate dot for either chromogenic or fluorescence detection. This highly specialized system utilizes signal amplification and background suppression methodology. RNAscope is compatible with a wide range of sample types and can be used to detect multiple RNA targets using one tissue section. Here's a brief overview of the RNAscope workflow. As I mentioned, we will go through each step in more detail later. RNAscope assay is a slide-based assay, and it is intended for use on sample types similar to that used with IHC or IF. The assay starts with the permeabilizing the sample with reagents provided in the pretreatment kit. The pretreatment allows for unmasking of the target RNAs, thus allowing the target probe to penetrate into the cell and hybridize to the target RNA sequence. The target probe is a pool of oligos that are designed using our established bioinformatics algorithm, which allows for target specificity. Once probes hybridize to target RNA, a series of amplification cascade steps can then amplify the signal by building an amplification tree. And RNA molecules can then be visualized under the microscope as punctate dots. The signal can also be quantified either semi-quantitatively or quantitatively using image analysis software. The first key feature of the RNAscope technology is the probe design. Each Z probe contains 3 elements. The lower region of the Z is an 18 to 25 base sequence that is complementary to the target RNA. Each double Z pair will bind approximately a 50 base pair region. The lower and upper regions are separated by a spacer sequence. The upper region of a single Z is a 14 base tail sequence. The upper 28 base sequence of the double Z probe is the preamplifier binding site, and it is where the amplification cascade occurs. For amplification to occur, 2 Zs must hybridize to a target sequence right next to each other. With a standard probe design, the target probe set is a pool of 20 double Z oligos pairs and is binds to a 1 kb RNA target region. Here is a schematic showing the hybridization and subsequent amplification of the RNAscope assay. The double Z probe will bind to the target mRNA sequence. Once the double Z probe binds, it serves as a binding site for the preamplifier molecule. Then the amplifiers are able to bind to the preamplifier, forming a branching complex at each DZ binding site. Each amplifier can further bind multiple labeled probes. These labeled probe molecules contain an enzyme, such as HRP, that generate a visible signal after chromogenic reaction, such as with DAB, that is detectable under a standard bright field microscope. The labeled probes can also contain fluorophores that allow for visualization of the signal under a fluorescent microscope. The signal amplification strategy provides for visualization of target RNAs as a single dot, where each dot represents the individual RNA molecule. The building of the amplification tree allows for increased sensitivity compared to traditional in situ. Further, if a single Z binds to the target mRNA transcript, the preamplifier cannot bind as it needs both ZZs to provide a binding site. This helps to ensure the specificity of the probe and helps reduce potential background staining. In order to detect mRNAs simultaneously, multiple amplification trees must be built. That being said, we must ensure specificity. The specificity is ensured by adding a unique tail sequence to the probes. With the double Z, within the double Z probe design, we have a tail sequence that is indicative of a channel designator. C is used to designate the tail channel for our RNAscope and BaseScope assays. T is used to designate the tail channel for our high-plex assays. This helps ensure specificity by ensuring the correct amplification tree is built for the correct probe channel. For example, a C1 probe has a specific C1 tail sequence that will only allow for binding of the subsequent C1 preamplifier, amplifier, and labeled probe. Similarly, a C2 probe will have a separate and unique tail sequence. The growth and adoption of the RNAscope technology is best exemplified by the number of peer-reviewed publications. Since their first publication in 2011, there have been over 2,700 peer-reviewed papers published in numerous journals, including many top-tier ones using the RNAscope technology. Publications using RNAscope have grown throughout the years, and the RNAscope technology is highly relevant across multiple fields of research, with over 40% of the publications in cancer research, followed by neuroscience and infectious disease. This slide summarizes all the assays that are currently available from ACD. We have Chromogenic single-plex brown or red assay and Duplex assay on both manual and automated platforms. The Multiplex fluorescent assay allows detection of up to 3 targets for the RNAscope fluorescent multiplex assay, or up to 4 targets for our V2 assay. The Hybridization assay can be used to detect up to 12 targets in a single tissue section. We also have some assays available on automation platforms, like the Ligabond RX or Ventana Discovery. For RNAscope, both the chromogenic singleplex and duplex assays are available on Leica and Ventana. The V2 fluorescent assay is currently available only on Leica. If your target of interest is shorter than 300 base pairs, it is below RNAscope detection limit. Currently, BaseScope is available on both manual and automation platforms for detection of targets between 50 to 300 base pairs. Now let's take a detailed look into the RNAscope manual assay workflow. Regardless of the RNAscope or BaseScope assay you are working with, you will follow this general workflow by gathering resources before you get started, preparing your samples, pre-training your samples to unmask RNA, hybridizing probes, and then amplifying, detecting, counterstaining, and mounting. We will begin by going over the resources you will need to get started. We recommend you download the specific documents necessary for your assay and sample type from our website. This will include an assay checklist to ensure you have all the necessary materials before beginning the assay. It will also include the user manual for your particular assay type and the technical notes for sample preparation and pre-treatment for your sample type. Some examples of sample types include FFPE, fixed frozen, flash frozen, cultured cells, and whole mass. Lastly, please contact us at technical support for additional tips or tricks if you have questions as to which user manual or technical notes are right for your application. The assay checklist will have a list of recommended materials that are specific to your assay. However, all of the assays will generally require superfrost plus slides, fresh reagents, an appropriate method for target retrieval, an image hydrophobic barrier pen, our Hybridization oven, control probes, control slides, tissue tech containers, appropriate mounting media for your assay, and lastly, the correct microscope and filter settings for your assay type. The RNAscope reagent kit comes with 3 subkits: the pretreatment reagents, the detection kit, and the wash buffer. You will also need target and control probes. Please make sure to warm your 50x wash buffer and probes at 40 feet for 10 to 20 minutes before making any dilution. When running the assay, allow the kit's reagents to come to room temperature before use. Here we have the Hybridization oven and Hybridization recommended accessories. On the left, the Easy Batch slide holder and wash tray will help for washing and handling slides. Our Easy Batch slide holder was designed to streamline your RNAscope assay and save handling time when working with a large amount of samples. The Hybridization hybridization system to the right is highly recommended since it is designed to maintain the perfect temperature and humidity to ensure optimal and consistent RNAscope staining. Here are some tips when working with the Hybridization oven. Allow the Hybridization oven to acclimate to 40 feet prior to use. Keep the humidifying paper wet throughout the assay. And additionally, the Hybridization oven can also be used for 60 feet baking steps. Next, we will discuss sample preparation. Sample compatibility is assay dependent. For instance, FFPE, fixed frozen, fresh frozen, cultured cells, and whole mount are compatible with the RNAscope singleplex, duplex, and fluorescent D2 assays. Our RNAscope fluorescent multiplex and hyplex assays are compatible with fixed frozen, fresh frozen, and cultured cells. Lastly, the BaseScope singleplex and duplex assays are compatible with FFPE, fixed frozen, fresh frozen, and cultured cells. This chart summarizes our recommendations for sample preparation, storage, and sectioning for FFPE, fixed frozen, and fresh frozen. For FFPE, we recommend using samples for 16 to 32 hours at room temperature using 10% NBS. FFPE blocks can be stored for 3 months at room temperature. For long-term storage of FFPE, we recommend blocks be stored at 4C. We recommend fixed frozen samples be fixed for 24 hours at 4C using either 4% PSA or 10% NBS. Fresh frozen samples should immediately be frozen after harvesting using either liquid nitrogen or dry ice. Both fixed frozen and fresh frozen samples can be embedded using OCT and stored at -80 for up to 3 months in an airtight container. For sectioning, recommended section thickness is dependent on sample type. For FFPE, we recommend 4 to 6 micron thickness. For fixed frozen, 7 to 15 microns, and for fresh frozen, 10 to 20 microns. For all, we recommend superfrost plus slides be used. Once you're ready to run the assay, part 1 of the assay is pretreatment. The pretreatment will vary depending on your sample type, your tissue type, and how the samples were prepared. Please note that the chromogenic-based assays, that is our RNAscope and BaseScope singleplex and duplex assays, as well as our RNAscope multiplex fluorescent B2 assay, have enzymatic-based detection and require an additional hydrogen peroxide step during pretreatment. One component of the pretreatment is the target retrieval. We have 2 options for target retrieval. Option 1 is using a hot plate where target retrieval is placed into the beaker and subsequently slides are placed into the target retrieval using a slide holder. Or option 2, using a steamer and placing a tissue tech container filled with target retrieval into the steamer. There are some important considerations for either of these methods. First, maintain the temperature between 98 to 102 degrees Celsius. To keep the target retrieval solution at a mild boil. Rapid boiling can cause tissue detachment. Lastly, please allow sufficient time for the target retrieval to reach temperature. This usually takes 20 to 30 minutes. It is important to plan ahead so that by the time you have reached the target retrieval step of your protocol, the solution is already between 98 to 102 degrees Celsius. Sample pretreatment also requires protease treatment. We offer 3 different proteases: protease plus, which is our weakest protease, protease 3, and protease 4, the strongest protease. The strength of protease be used and duration will depend on a few factors such as sample type, for instance, is it FFPE, fixed frozen, or fresh frozen, your tissue type, some tissues require milder or stronger protease, and lastly, how the samples were prepared, were they underfixed or overfixed, for example. The protease step requires the use of the high-easy oven and high-easy humidity tray. Please ensure the high-easy oven is turned on at least 30 minutes prior to use so that it is acclimated to 40°C. For fresh frozen samples, protease pretreatment is performed at room temperature and not at 40°C. Shown on the slide are sample pretreatment recommendations for various sample types to perform RNAscope. The recommendations shown on this slide are for manual assay platform. With each sample type, we are varying the target retrieval time and protease treatment. For FFPE, we recommend 15 minutes of target retrieval and the use of protease plus for digestion. For fixed frozen, we recommend 5 minutes of target retrieval and a stronger protease, protease 3, is used. However, for fresh frozen samples, target retrieval is not required, and hence the strongest protease, protease 4, is used. Lastly, for culture cells, again, no target retrieval is required, and a dilution of protease 3 is recommended. Please note that BaseScope assays may require a stronger protease pretreatment than RNAscope. Sample pretreatment can also depend on tissue type. Some tissues require a milder pretreatment, and some tissues require a harsher, extended pretreatment. This chart shows mild, standard, and extended pretreatment times for FFPE samples. For a list of tissue-specific pretreatment recommendations, please refer to the user manual appendix. Once the pretreatment unmasks RNA, the RNA is accessible for probes to bind. Next, we will discuss probe hybridization. The conditions for probe hybridization are universal for all of our assays. Our C1 and pooled control probes come ready to use in a dropper bottle at 1x. All other probes will come as 50x concentrates. For a single-plex RNAscope or BaseScope assay, you will need a C1 probe. RNAscope or BaseScope duplex assays will require C1 and C2 probes for detection of the two targets. The table summarizes the probe channels for each assay. The probe hybridization step requires pooling of all channel probes for the assay. 50x probes can be diluted using the C1 1x probe. If you would like to avoid C1 detection, 50x probes can be diluted using the probe diluent. High-plex probes T1 through T12 are all at a 50x concentrate and require dilution into probe diluent. It is important that probes are placed in a 40C water bath for 10 to 20 minutes prior to making any dilutions or before using for probe hybridization. For all of our RNAscope and BaseScope assays, probe hybridization will be carried out at 40C using a high UV oven for 2 hours. Following probe hybridization, there are two options. There is an optional stopping point after hybridization, so the assay can be broken up into 2 days. If stopping, slides can be placed into 5x SSC buffer overnight and left at room temperature. It is important to add enough SSC so slides do not dry out overnight. The next day, you can continue with the assay by washing twice with 1x wash buffer and proceeding to AMP1. After probe hybridization, option 2 is to continue to amplification steps. If you plan to continue the assay on the same day, I would recommend that the detection kit be placed at room temperature during the hybridization steps to allow sufficient time for the reagents to acclimate to room temperature. For additional stopping point information, please reach out to us at technical support. Next, we will take a detailed look into the steps required for building of the amplification tree and detection. These steps are assay dependent. First, we will take a look at the amplification and detection of the RNAscope single-plex red and brown assays. After probe hybridization, there are subsequent AMP steps. For all RNAscope and BaseScope assays, please note that the times and temperatures vary during these steps, and it is important that these conditions be followed. For instance, in this case, AMP5 and AMP6 requires that they be carried out at room temperature and not at 40C. For all RNAscope and BaseScope assays, it is important to carry out all of the AMP steps and in the correct order. For detection, the red assay utilizes ALP and fast red chromogen. Please note that the fast red chromogen is alcohol sensitive. The brown assay chemistry is HRP based and uses chromogen DAB. The RNAscope duplex assay has sequential detection of red and green chromogen. Following AMP1 through AMP6, you will detect the red chromogen first using ALP fast red chemistry. Please note the red chromogen detects the channel 2 probe. After red detection, AMP7 through AMP10 are required for green detection. The green chromogen detection is HRT based and will detect channel 1 probe. Both the fast red and green chromogens are alcohol sensitive. The green chromogen is also water sensitive. The RNAscope Fluorescent Multiplex requires 4 AMP steps that are all carried out at 40C. AMP 1 through 3 will build the amplification trees for all the probe channels. Then AMP 4 step will place fluorophores onto these amplification trees, allowing for detection. There are 3 different choices for AMP 4, allowing for 3 different color module configurations for which fluorophore is used for detection of each channel. For Alt-A, Alexa 488 detects C1. For Alt-B, Alexa 488 detects C2. And for Alt-C, Alexa 488 detects C3. Here is a depiction of the AMP4 FL Alt-A module configuration for each channel. Here, Alexa 48 detects channel 1, 80550 is used to detect channel 2, and 80647 is used to detect channel 3 target. Next, let's look at the amplification and detection workflow for the RNAscope Multiplex Fluorescent V2 assay. AMP1 through AMP3 for V2 assay builds the amplification tree for all 4 channels. Following the building of the amplification trees, there is a sequential detection of the targets. Here we have a depiction of the detection of the first channel, which requires the binding of HRP-C1 to the C1 amplification tree. Then the addition of an opal or TSA plus dye is added. The opal dye will react with the HRP-C1 to become active by forming a radical. This active dye will rapidly react and deposit a fluorofluor. The V2 assay allows for flexibility, as any opal dye can be assigned to any channel. The opal dye concentration can also be optimized, thereby allowing for the ability to control the intensity of the staining. We recommend starting with a 1 to 1500 dilution of opal dye, with a recommended range of 1 to 750 to 1 to 3000. Once the opal dye is added, the HRP reaction is blocked with the addition of HRP blocker. After HRP-C1 is blocked with the HRP blocker, the next HRP, HRP-C2, is added, and channel 2 is detected with the addition of another opal dye. Then HRP-C2 is blocked with the HRP blocker. This is repeated for detection of channel 3 and channel 4 targets. The high-plex assay, which allows for the detection of 12 targets, requires building of all 12 amplification trees by the addition of AMP1 through AMP3 steps. Following the building of amplification trees, there is a sequential detection of the 4 targets at a time. First, fluoro T1 to T4 are added for detection of T1 to T4 targets. The sample is then counterstained and mounted for imaging. Following imaging of round 1, the cover slip is removed. The T1 to T4 fluorophores are cleaved off using cleaving solution. Then T5 to T8 are detected using fluoro T5 to T8. The samples are again mounted and imaged for round 2 of the staining. Following imaging of round 2, the cover slip is again removed and fluorophores detecting T5 to T8 are cleaved off using cleaving solution. Then fluorophores T9 to T12 are added for detection of the last round of targets, T9 through T12. The slides are counterstained for dapping and mounted for the last round of imaging. Now, the high-plex assay uses the high-plex image registration software to overlay the 3 rounds of images. The BaseScope singleplex assay requires 8 sequential AMP steps. The AMPs vary in temperature and duration, so it is important that these steps be followed accurately. The BaseScope singleplex red assay involves ALP fast red chemistry for detection of the C1, that is channel 1 target. The BaseScope duplex assay, similar to the RNAscope duplex assay, has sequential detection of the red and green chromogen. Following AMP1 through AMP8, the channel 2 target is detected first using the ALP fast red chemistry. After red detection, AMP9 through AMP12 are required for green detection. The green chromogen detection is HRP-based and will detect the C1 channel 1 target. Lastly, we will discuss counterstaining and mounting your samples. We recommend 50% Gills hematoxylin 1 for counterstaining of the RNAscope and BaseScope singleplex and duplex chromogenic assays. For fluorescent-based assays, DAPI is provided in the kits for nuclei counterstaining. We recommend cytosol XYL for the singleplex brown assay. Either Vectamount or EcoMount are compatible with our RNAscope singleplex red, RNAscope duplex, BaseScope singleplex, or BaseScope duplex assays. Lastly, for our fluorescent-based assays, we recommend the use of prolonged gold anti-fade for mounting. Please note, the prolonged gold anti-fade mounting media requires time to cure before imaging. I will now turn it over to Adeola for the second part of this presentation.
Thank you, Anisha. We will now move on to the second half of the presentation, which will cover the best practices to follow while running RNAscope assays and analyzing the results. A best practice that we highly recommend is to include controls, both positive and negative, in your assay. Positive controls are used to ensure that the assay workflow is correct and to confirm RNA integrity in your sample. Negative control is used to confirm signal specificity. Duplex assays use PPID as a positive control and DAP-B as negative control. PPID is a housekeeping gene, and most normal tissues are expected to show moderate PPID expression, while DAP-B is from a cell bacterium and should not pick up any signals in samples from other species. For duplex assays, we recommend a duplex positive control with polar 2A and PPID, and a duplex DAP-B negative control. For multiplex assays, we recommend a 3-plex or 4-plex positive control comprising of polar 2A, PPID, UBC, and HBRT, and a negative control with 3-plex or 4-plex DAP-B. Lastly, the high-plex assay includes 12-plex positive control, which is species-specific, and 12-plex DAP-B negative control probes. Listed in this table are the housekeeping genes that make up the mouse 12-plex positive control probe. If you will be performing the RNAscope assay for the first time, then we recommend that you perform a technical quality control using the positive and negative control probes on control slides that are available from ACD. These are FFPE, HeLa, or 323 cell pellets. This will confirm that the experiment setup in your lab is optimal and the assay is being run correctly. If the control slides pass QC based on our scoring guidelines, which I will describe in a later slide, then you can proceed to qualify your own samples. By running positive and negative control probes on your samples, you will confirm that the samples have been optimally prepared, that RNA quality is preserved, and you will determine if the standard assay conditions are the best for target detection. If your sample passes QC, you can then move on to running your target probes. Otherwise, it is recommended that you obtain new samples with good RNA quality, or you perform optimization of the assay protocol. Please feel free to contact ACD Technical Support, and we are more than happy to help you with assay optimization. Here is an example of RNA quality validation for the duplex assay, where cells are stained with our duplex positive control probe or negative control probes. In these images, you can observe fairly uniform expression of the positive control housekeeping genes, PPID, and polar 2A, indicating good RNA quality. Conversely, the DAP-B section is clean with no signal or background. The next two slides are examples of successful staining using our 4-plex positive control and negative control probes on control slides with a multiplex fluorescent V2 assay. With any new sample you're working with, we always recommend running positive and negative control probes to assess the sample RNA quality and the assay conditions. Here are the images for the 4-plex negative control slide. No signals are observed in any of the channels and the background is clean. On this slide, we have the same samples and assay as before, but stained with the 4-plex positive control probe. Here, polar 2AC1 is detected with opal 520 dye, PPID-C2 with opal 570, UBC with opal 620, and HPRT with opal 690. By running the 4-plex control, you can confirm RNA quality and ensure that you're observing the expected staining for each housekeeping gene with the assigned fluorophore, and that the imaging and filter settings are optimal. Next, we have an example of successful staining using our 12-plex positive control probes on mouse hippocampus with a high-plex fluorescent assay. These are individual channel images which can then be compiled using our high-plex image registration software to view all 12 genes together. Here's an example of a typical RNAscope experiment. Serial sections from a human lung cancer sample were used in the manual chromogenic red assay. On the left is a section stained with a negative control probe, DAP-B, and as observed in the image, there should be no signal from DAP-B. In the middle panel is a section from the same sample stained with a positive control probe, PPID. The PPID signal is strong and is fairly uniform across the section, indicating that RNA integrity is good in the sample. Certain tissues may express the positive control probe at higher or lower levels, but in general, you want to see expression throughout the sample. With the results from the first two sections, we can have confidence in the target probe imaging in the far right panel. This panel is a section from the same lung cancer sample probe for the image from the immune checkpoint marker PD-L1. PD-L1 exhibits a wide range of expression in tumor tissues. Here you can observe strong punctate dots with the PD-L1 probe indicating expression of PD-L1 in this tumor sample. So you can conclude that the localized pattern of expression we observed for PD-L1 is in fact correct. After successful detection of your RNA targets, you may be interested in characterizing cell type specific expression by visualizing cell surface markers with your RNA of interest. The RNAscope assays highlighted here in red can be complemented by IHC or IF, the simultaneous RNA and protein detection on the same slide. What we refer to as a dual ISH-IHC or IF assay can be used to visualize RNA binding proteins and their target RNA and to dissect the regulation of gene expression. We have detailed technical and application notes available on our website with guidelines for combining your RNAscope protocol with protein detection. You can also contact us at technical support for that information. Moving on to determining the optimal conditions for your samples. Here are some potential reasons for why you may need to optimize your assay conditions. If the samples are underfixed, this can usually lead to overdigestion of your samples using standard conditions. If, for example, they're fixed for less than 16 to 32 hours as is recommended for FFPE samples, then you may need to optimize the conditions. If the samples are overfixed, the signal could be weak and you may need to optimize to improve signal detection. There are also particular sample types that may require different pretreatment conditions and optimization. A few are listed here. For example, bone samples will usually require decalcification, retina will require milder pretreatment, and liver and muscle may require extended pretreatment. If your tissue of interest is not listed here or you're unsure if your tissue has been fixed as recommended, please feel free to reach out to technical support for guidance. Here's an example from a sample that was fixed for too long and then stained with the singleplex brown assay. In the image on the right, you can observe that the RNAscope signal intensity is very weak compared to the left image where the sample was fixed as recommended. This shows how important it is to prepare and fix your samples per our recommendations. Additionally, the fixation state of your samples will be important in determining the optimal pretreatment conditions. Sample pretreatment is critical for the success of RNAscope. When running the RNAscope assay in your samples, begin with the recommended conditions for your sample type. Observe for signal intensity and background with control probes and sample morphology. Unless otherwise noted in our user manuals or tech notes, it's always best to start with standard conditions. This will help to set a baseline for how the assay performs, and based on your results, we can make recommendations to either scale the pretreatment up or down. Both target retrieval and protease digestion can be performed in a sliding scale, such that you may adjust them depending on the sample preparation and fixation conditions of your samples. This slide shows optimal digestion of a sample that was fixed according to ACD's recommendation and treated with the standard pretreatment conditions. On the left, the PPIB staining is strong, while the DAPB negative control slide on the right has no signal. You can also see that the tissue morphology is intact and the hematoxylin staining is homogeneous. Now let's review some troubleshooting examples and the results after performing optimization on these samples. Compared to the previous slide, this image shows tissue that has been underfixed, which led to overdigestion in which the nuclei are now undefined due to suboptimal tissue morphology. Here, milder pretreatment conditions are needed by reducing the target retrieval time, and this will improve the morphology and eliminate background staining with the negative control probe. Similarly, there is better morphology and higher signal resolution with the modified target retrieval time in underfixed sections stained with the control probe PPIB. Overdigestion from protease pretreatment can also affect fluorescent RNAscope signal quality. With the mouse kidney sample shown in the top panel, you can see there is quite a bit of tissue autofluorescence on the left when protease digestion is performed at 40 degrees. The tissue is overdigested, and not only do you not see any punctate signal, there is also an increase in background. But when the tissue is appropriately digested using protease 4 at room temperature in the right panel, distinct green and red dots can be observed with the control probes. Similarly, as shown in the bottom panels, with adequately treated fresh frozen mouse brain samples, there is no background and you can observe beautiful punctate signals. And with tissue digestion, you can see an increase in background and a decrease in signal. On the opposite end of the scale, here are examples that have been underdigested. The PPIB staining is much weaker, but the tissue morphology looks great with strong hematoxylin, and it shows no signs of overdigestion. To account for this, the target retrieval was adjusted from standard to extended, which improved signal intensity and resolution and eliminated the nonspecific trapping of the chromogen, which is seen in the image on the left. Another issue you may experience is related to tissue adherence to slides. To troubleshoot sample detachment, we recommend using SuperFOS Plus slides to ensure optimal adherence. Tissue detachment can also be caused by suboptimal sample preparation. We have found that including an additional baking step can mitigate this issue. I will now review in detail the additional baking steps to be incorporated for FFPE and fixed frozen samples. These will apply to all assays. If you're working with FFPE samples on observing tissue detachment, we suggest adding a baking step before hydrogen peroxide, or if you're observing tissue coming off the slice after protease treatment, add an additional baking step before that. Both baking steps should be performed for 30 minutes at 60 degrees. If sample detachment is observed with fixed frozen tissues, we recommend drying the slides at minus 20 degrees for 2 hours after cryosectioning and adding a post-fixation step before hydrogen peroxide for 15 minutes to an hour at 4 degrees. We have found that majority of tissue detachment happens after the target retrieval step, so we recommend adding an additional baking step before protease treatment. If after trying steps 1 to 3, you continue to observe tissue detachment, then we recommend that you omit the target retrieval step altogether and extend the protease time to 45 minutes. You can also contact technical support and we will help you troubleshoot the best conditions for your tissue. I will now discuss how best to capture high-quality images for subsequent data analysis. Here are a few general guidelines to follow when acquiring images from your RNAscope assay. Consult with your imaging or microscope core labs to determine the optimal settings for your instrument. Run a positive and negative control slide in each experiment for comparison. Always use correct filter settings for the fluorophores on your section. Image the slides within 2 weeks of fluorescent staining. Next, we will discuss some challenges that you may encounter when acquiring images from your RNAscope assay. Let's start with bright field imaging for chromogenic assays and talk about false signals. The images on the left are the negative and positive control probes that you would have seen in an earlier slide. We recommend that you always include negative and positive control slides as they provide a point of reference for your results. The images on the right are representative images that may give a false signal. The first is an image with a pink haze that is inherent to the tissue, which may appear similar to the color in our red assay. But in a high-quality image, as seen on the far right, we are able to distinguish the colors between this pink haze and the red signal based on their intensity. There are other structures and tissues which may have colors, like the example seen here with tar, lactophysin, and melanin. These are easily distinguishable from red color, but they may be difficult to separate from brown signal. So for these kinds of tissues, we would not recommend using the brown chromogenic assay. Next, we will discuss fluorescence imaging. One of the main challenges in fluorescence imaging is tissue autofluorescence. Here you can see the autofluorescence in different tissue types. Autofluorescence is intrinsic to biological specimens and should be excluded during image analysis. However, this may not always be easy as it may be difficult to separate it from real fluorescence signals. One way to distinguish if a signal comes from autofluorescence is to check if the same signal is present in different channels. because autofluorescence usually has a wide spectrum, which makes it visible through more than one channel. Here is a checklist to review and to use to determine whether or not your image is of good enough quality to continue with analysis. Make sure your images are in focus. For chromogenic images, make sure the colors are well separated. For fluorescent images, ensure the fluorescent channel read-through is minimized. Minimize autofluorescence. Confirm that the signal is not saturated. And lastly, review reference images for positive and negative control probes, which are available for most combinations of tissue, type, and assays. Moving on to the methodologies available for analyzing your RNAscope images. When analyzing RNAscope data, it is important to remember that you can expect to have higher quality data if you start with good quality images. As the saying goes, garbage in equals garbage out. In general, RNAscope data analysis methods can be divided into two major categories: semi-quantitative or quantitative analysis. We have data analysis guidelines available for both methods. For most studies, the approach of counting ISH dots and calculating the number of dots per cell will be the most appropriate. However, there is no one universal method, and each researcher will need to determine the right method for their specific research question or goals. Some frequently asked questions we get from customers include, what is a typical RNAscope signal? What does a dot mean? Does dot size really matter? Why do I sometimes see a cluster versus a dot? So we'll address these questions next. In this image from a chromogenic red assay, the signal appears as numerous punctate red dots of various sizes and intensities and a few clusters. It is the number of dots and or clusters that are important, rather than the size of the dot or the intensity of the dot. Each punctate dot, as shown by the blue arrow, represents one mRNA molecule. The slightly larger, darker red punctate dot, shown by the yellow arrow, is also one mRNA molecule. However, it is larger in size as more number of ZZ probes have bound to the target molecule. In contrast, a cluster shown by the brown arrow is oblong with a slightly irregular border and is the result of overlapping signals from multiple mRNA copies. Similar patterns of dots and clusters are seen within the multiplex fluorescent assays. The yellow arrows point to single mRNA molecules with fewer ZZ probe pairs bound to them, compared to the dot shown by the teal arrow. The white arrow is pointing to a cluster with an irregular shape due to overlapping signals from multiple mRNA molecules. RNAscope staining can be quantified by manually counting the number of dots per cell. This is known as a semi-quantitative scoring method. It is a visual assessment that provides expression levels based on the percentage of cells that are positive. With this method, the cells are binned according to the number of dots per cell, and we score the samples from 0 to 4 based on the average number of dots per cell. For example, if you see 4 to 9 dots per cell with no or very few clusters, that is a score of 2. The score of 2 is what we would like to see with positive controlled PPIB staining on most samples during sample QC. This same scoring guideline is also applicable to images from the multiplex fluorescent assays. For quantitative methods of RNAscope analysis, there are many vendors that offer software that can be used to analyze your images. The two popular open source options that we recommend for RNAscope or BaseScope are ImageJ and Cell Profiler. If you would like to get started using these, please download the most recent versions via the links listed here. When performing data analysis, an important document that we encourage you to review is a reference guide which has been developed by ACD scientists with examples of different scenarios of RNAscope staining and explanations on how data can be analyzed in each scenario. This document is called A Guide for RNAscope Data Analysis and can be downloaded from our website. Additionally, there are technical notes available which provide step-by-step instructions on how to quantify RNAscope or BaseScope using either ImageJ or using Cell Profiler software. The last section of the presentation will review the resources that are available to help you perform RNAscope successfully. Please visit our website at acdbio.com for more information about our products and the resources available to you. There you'll find a wealth of information, and if you go to the support page, there are user manuals for the manual and automated assays, as well as troubleshooting guides and tech notes. You can also review training videos for different steps in the assay workflow, as well as previously recorded webinars that cover a wide range of topics and applications related to RNAscope or BaseScope, including our probe design webinar, which provides an overview of probe design strategies and instructions for completing our online new probe request form. A BaseScope assay webinar, which details the wide applications of the BaseScope assay for detection of exon junction, SNPs, and point mutations. We also have a data analysis webinar, which has a live demo of using ImageJ to score RNAscope images, and a cell profiler training webinar, which describes the cell profiler image analysis workflow and also includes a live demo. Another important resource available to you is our new user program. When you are enrolled in this program, you will be paired with an ACD technical support scientist who will work with you directly to provide guidance on the following steps: your experimental design, guidelines on how to get started with your assay, sample preparation and pretreatment recommendations, assay workflow and interpreting your results. And lastly, we'll also provide troubleshooting tips. On the new product side, we now have a new assay platform to detect microRNAs called miRNAscope, which will become available soon. miRNAscope is a robust assay offering high sensitivity and high specificity with a universal probe design. The custom probe is designed to detect microRNAs, antisense oligonucleotides, or small interfering RNA sequences of interest. Like RNAscope or BaseScope, the miRNAscope assay can be combined with downstream protein detection and is compatible with FFPE, fresh frozen, fixed frozen, and cultured cell samples. Initially, it will be available as a single-plex chromogenic fast-read assay on the manual and microautomation platforms. Please take note of our technical support contact information, and feel free to reach out to us if you have any questions or if we can be of any assistance. With that, we have come to the end of today's presentation, and I'd like to thank everyone in attendance today. We appreciate your patience through our technical difficulties. Some of the most frequently asked questions will be posted next, and our colleagues are still on the line to help you address questions. Please continue sending your questions in. As a reminder, this webinar will be recorded and available on the website shortly.